this video lecture, we will discuss about method of studying soil microflora. We will see different method for studying soil microflora. Okay. First, we will see qualitative analysis. The term qualitative aim to estimate the number of soil microflora. Quantitative, that is, always deals with the number. We can come to know the numbers. Technique that are involved such as SPC, that is known as standard plate count technique. Second, that is direct microscopic technique. And the third, that is determination of MPN, that is most probable number. These all techniques that give the idea about the number of microorganisms that are present in the soil. B, that is qualitative analysis. It includes various methods for isolation of specific organism from soil. For example, isolation of rhizobium. It is a symbiotic organism. It has a symbiotic association with the leguminous plant for the nitrogen fixation. Isolation of azotobacter. It is a non-symbiotic organism. Isolation of actinomycetes, fungi, yeast and phages. First, that is the collection of sample. For method of studying the soil microflora, first thing that we have to collect a sample. Soil sample are collected normally at the depth of 15 centimeter. At the depth of 15 centimeter, we collect the soil and transfer into the clean container or sterile container. From the 15 centimeter depth, we collect the sample and we transfer into the clean container. Once the sample collection is done, then we can go for various method or various technique. Various methods for studying the soil microflora are direct macroscopic method, buried slide method, enrichment culture technique, agar plate technique. These all are the different methods for studying the soil microflora. We will see each and every method in detail. First, we will discuss about direct microscopic method or examination of direct microscopic. In that, first of all, we will take a 1 gram of soil from the whatever region, from the whatever the place we have taken the soil. From that soil, we will take the 1 gram of soil and we transfer into the 9 ml of 0.0015% agar solution, that is nutrient agar solution, and mix it. After that, we take 1 ml of suspension. From above mixture, we will take 1 ml of suspension to the center of slide. We take a slide. In the slide, from the above section, we will take a suspension of 1 ml and suspension is evenly distributed, and we will evenly distribute the suspension and we air dry the slide. After the air dry, fix it with 0.1 normal HCl for a minute. For a 1 minute, we will fix it with 0.1 normal HCl. It is a chemical fixative. After that, then immersed in water. To remove excess water, we immersed in water. Again, we air dry, and after air dry, we will stain the slide with rose bengal or erythrocin. With the help of stain such as rose bengal or erythrocins, we will stain the slide. After staining the slide, again we will air dry and we will examine under the compound microscope. In the direct microscopic method, first of all, we will take 1 gram of soil and we will add into the 9 ml of 0.0015% of agar solution and we will mix it well. After mixing well, now the suspension is ready. From that, we will take 1 ml of suspension and we will make a smear with the help of nichrome wire or with the help of pipette we will make a smear. Then the suspension we will allow it to air dry. After air dry we will fix it with 0.1 normal HCl. It is a chemical fixative. Then we will immerse into a water to remove the excess acid. Again we will air dry and we will stain with rose bengal or erythrocin stain. And again we will air dry and we will examine under the compound microscope. For quantitative analysis, counter chamber is used. Example, Petrov-Hausser counting chamber. This is what all about the direct macroscopic method. This method was originally discovered by Kohn. In the year 1918, it was first discovered by Kohn in 1918. Now the second method, that is buried slide technique. It is an old method. Buried slide technique method, it is an old method. In this method was first discovered by Rossi and Colandi. Make a slit in a soil with sterile knife. We will make a hole with the help of sterile knife into the soil and introduce the clean glass slide in slit. Whatever the slit that we have prepared, with the help of sterile knife, we will introduce our clean slide into this slit and we will leave it for 1 to 3 weeks in the same position. Whatever the position that we have inserted, in that position we will leave it for 1 to 3 weeks. After 1 to 3 weeks, we will remove the slide and wipe the slide from one side. On the one side we will gently wipe one side and wash the slide with water, and again then we will air dry and heat fix over the flame. After air dry we will heat fix the slide with the flame and allow it to air dry and examine under the microscope. This is what all about the buried slide technique. In this, first of all we will make a slit with the help of sterile knife. In that, whatever the slit that we have made, we will insert a glass slide into this slit and we will leave it for 1 to 3 weeks at the same position. After 1 to 3 weeks, we will remove the slide and from the one side we will gently wipe it, whatever the soil has been attached. Then we will wipe and wash, and in the another side the soil will be attached onto the one side of the slide. In that we will air dry and heat fix over the flame, allow it to air dry and we will examine under the microscope and we will count. This is what all about the buried slide technique. It is an old method. Third, that is enrichment culture technique. In the enrichment culture technique, this technique was first developed by Martinus Beijerinck. Enrichment culture technique, it was first discovered by Martinus Beijerinck. Enrichment culture is the medium that favor the growth of particular microorganism. Enrichment, it is that enrich the growth of microorganism, that favor the growth of microorganism, that kind of medium that we will use. It will support the growth of selected microorganism. Enriched, it will selected for the microorganism. Example, if you want to isolate the halophiles that can only grow in the presence of high salt concentration, the media containing high salt concentration, that will favor the growth of halophiles and we can get the halophiles organisms. Selenite broth, it is a selective for Salmonella species. Selenite broth, that is selective for the Salmonella species. Alkaline peptone water, it is used for the cultivation of Vibrio. These all are the selective media for the particular microorganisms. Selective media, or we can say enriched media for that particular microorganism. In the heterogeneous inoculum, such as soil is an heterogeneous in which various microorganisms are present, diversified organisms that are present inside the soil. So, in the heterogeneous inoculum such as soil added into the liquid medium, that is the enrichment medium, from this we will take a soil sample, we add into the enrichment, specific enrichment medium. Microorganisms will adapt in this condition, in that liquid medium, enriched medium, in that condition will grow and start proliferate, and they will start growing and proliferating, and it will then, and it if transferred into the fresh medium, obtain a pure form, and again we transfer into the pure fresh medium to obtain in a pure form. This is what all about the enrichment culture technique. In the enrichment culture technique, it is a medium. Enrichment culture is a medium that favor the growth of particular microorganisms. We will take a soil sample and we will transfer the soil sample into the enrichment medium. From the enrichment medium, the microorganisms will, only the microorganism that is enrichment, for that they will adapt and proliferate and grow. And from that again we will transfer into the pure medium. Agar plate technique. In the agar plate technique, it is mainly used for the isolation and enumeration of bacteria, yeast and mold present in the soil. It is mainly used for the isolation and enumeration of bacteria, yeast, mold and that are present in the soil. For the agar plate technique, first of all we will take the 1 gram of soil. Then we will add into the 10 ml of distilled water, sterile distilled water. We will add 1 gram of soil into the 10 ml of sterile distilled water tube. Mix it well and allow it to stand for few minutes. We will mix well and we will allow it to stand for few minutes. After that, we will prepare a serial dilution of soil sample. Serial dilution of 10 raised to minus 1, 10 raised to minus 2, 10 raised to minus 3 and so on. We will prepare a various dilution. From the 10 ml of suspension, we will prepare a various dilution. After the serial dilution, from each of the dilution tube, we will transfer 0.2 ml, we will withdraw 2.2 ml and we will transfer into the sterile melted agar. In the melted agar we will add. After adding into the melted agar tube, immediately we pour into the sterile petri plates. Sterile empty petri plate we immediately pour. After that, after pouring, we allow it to solidify. Once it gets solidified, incubate all the plates at 37 degrees Celsius for 24 hours. After that 24 hours, colony will be formed. We will count the total number of colonies that develop on each plate. Whatever the plates we have incubated for 24 hours, on the next day we will take out the plates and we will count the colonies on each plate. In the agar plate technique, first of all, we will take 1 gram of soil and we add into the 10 ml of sterile distilled water and we will mix well. After mixing well, we will allow it to settle down for a while. Then we will prepare a various dilution. After preparing the various dilution, from each of the dilution we will take 0.2 ml and we will add into the melted agar tube. In the melted agar tube, then we immediately pour into the empty petri plate, sterile empty petri plate, and allow it to solidify. Once it gets solidified, incubate all the plates at 37 degrees Celsius for 24 hours. On the next day, we will count the colonies that are present on each plate. It is, if necessary, use colony counter to help the colony counting. Whatever the colony that are present in the plate, we can count with the help of colony counter. If colony is between 30 to 300 are countable plates. If it is less than 30 or more than 300 are not acceptable. We will only consider between the colonies between 30 to 300. Now we will see what are the advantages. Total viable cells can be obtained. Disadvantages is obligate anaerobes cannot grow. Autotrophic bacteria cannot grow. cellulose decomposer fail to grow, sulfate reducing bacteria cannot grow in the absence of sulfate. This is what all about for method of studying soil microflora.